Good afternoon. I'm Bud Baslak, the Provost and Executive Vice President here at Case Western Reserve University, and it really is my privilege to welcome you to the 7th year of this outstanding program, the Ford Distinguished Lecture Series. We've hosted people like Jeff Hawkins, the inventor of the Palm Pilot, Bob Langer, a leading scholar of tissue engineering, and Dean Kamen, the developer of the Segway. Every guest brought to campus through this program does more than deliver a talk like we're going to experience here in a few moments. Each also spends time with the faculty and students in our Department of Biomedical Engineering. Our community has the opportunity to learn firsthand from these pioneers of innovation. At the same time, these pioneers get to see our faculty and begin to understand why biomedical engineering is among the most highly regarded programs here at Case Western Reserve University. Their ideas and inspiration came to Case Western because of the vision and passion of two remarkable people, Alan and Constance Ford. As you can see, Alan is here with us in person. Alan, it's gotten dark, but perhaps you can still wave. There he is over in the corner. And he's always at these events. Let's see, now, let me take a moment here. Somebody is standing next to the light switch. So, all right, we're good. Alan's belief is that the mission and potential of our great Department of Biomedical Engineering has kept him involved as one of our 4 greatest thought leaders, supporters, and at times biggest fans. He recognizes the immense talent of our faculty, many of them are here today with us, and their unparalleled dedication to making a positive difference at the university and certainly to our students. Alan joined our Board of Trustees in 1976 and chaired the board from 1987 to 1992. He's a recipient of the University Medal, which is the single highest honor that Case Western Reserve University bestows. And he continually is engaged as an emeriti trustee with that board, and actually our board meeting is this weekend, so he'll be busy at that. Alan, again, for all that you do on behalf of the program, the university, let's have one more round of applause. Thank you. And now it's my great pleasure to introduce to you the interim chair of the Biomedical Engineering Department, Professor Bob Kirsch. Bob.
Wow, this is great to see you all here this afternoon. Thanks for coming. As Bud said, I'm Bob Kirsch. I'm the interim chair of biomedical engineering. And before I introduce our Ford lecture today, I want to echo Bud's comments about you, Alan. You've been a great supporter of our department for many years. We're continually grateful for all your advice and support. So thank you very much. I'd also like to thank Jeff Capadona. Jeff is the organizer of today's and in case you didn't hear the news, Jeff was a recent recipient of a Presidential Early Career Award for Scientists and Engineers. So, PK, this is very prestigious. So again, I'd also like to thank Stathis Karathanasis, who has helped Jeff organize this. Lauren Biddlecombe, who's put up with all this stuff in the last few minutes. And Ann Borchard for doing a lot of the legwork for putting the event together. You should mark your calendars for our next Ford lecture, which is in February, and it's going to be Rod Pettigrew, who's the director of the National Institute on Biomedical Imaging and Bioengineering. So that's the spring lecture, but it's in February 19th. So I guess we'll call that early spring. So it's my great pleasure to introduce Chad Mirkin from Northwestern University as our Ford lecturer today. So Dr. Mirkin is the George R. Rathmann Professor of Chemistry. He's Professor of Medicine, Material Science and Engineering, Biomedical Engineering, and Chemical and Biological Engineering. And he's the Director of the International Institute for Nanotechnology and the Center for Nanofabrication and Molecular Self-Assembly at Northwestern. So his research, which we'll hear about today, he's known for the development of nanoparticle-based biodetection schemes, the invention of dip-pen nanolithography, and contributions to supermolecular chemistry, nanoelectronics, and nano-optics. So Chad is really accomplished. So he has more than 500 publications, 500 patent applications, and more than 200 of these have been issued. For the 10 years ending in 2010, he was the most cited chemist, the second highest impact factor for a chemist, and the most cited nanomedicine researcher. His discoveries have led to more than 50 commercial products, He's been awarded an incredible number of honors and awards. He's a member of the National Academy of Science, the National Academy of Engineering, and the Institute for Medicine. So these are all three of the national academies. He had an NIH Pioneer Award. He has honorary degrees from several universities in the U.S. and internationally. I heard about Singapore earlier today, where he has a lab, actually. He's on President Obama's President's Council of Advisors on Science and Technology, which is also quite a big deal. So I think I could read for the next hour with Chad's accomplishments, actually, and kind of you voted with your feet here today. So I'm going to let him introduce his own title. I'm sure that's going to resonate with a lot of us. Our department does a lot of nanomedicine activities. We have drug delivery, imaging contrast agents, molecular imaging, surface modification for biomaterials, nanoscale structure function analysis of orthopedic biomaterials. Chad's also very translational. We have our Coulter Translational Research Partnership, where our faculty work with individuals in the School of Medicine and others to turn our discoveries into new products. And I hope we can be as successful as you've been, Chad. So we're all looking forward to hearing your talk today, and let me welcome Chad Mirkin.
Okay. Thanks for a really great introduction, and thanks to all of you for coming here to hear a little bit about some of the things that I'm really quite passionate about. I also want to send my thanks to Mr. Ford, who's been very generous, obviously, to set this opportunity up. I think these are really quite important. We try to run several of these at Northwestern, and to have people from the community step up and help sponsor them is absolutely critical to, I think, not only improving the way we learn at the university, but to reach out to the rest of the community and give them a feel for some of the important things that are going on within the universities. I'm going to try to use today to give you a feel for what I think is important in the field of nanoscience, a field that is admittedly relatively new. The modern aspects of it have only been around for a little over a decade. Been the talk of science fiction for quite some time, and I think it's reasonable to begin to ask, what is this all about? What are the opportunities within it, and what has been accomplished to date? And I'm not going to give you a total view of the entire field. I'm going to give you some Mirkin-centric views and Northwestern-centric views, because those are the ones that I'm most familiar with, and I think they illustrate, some of the potential of the field. Do we have a problem here? We may. For some reason, this is locking up again. Where's our student there? Good. Okay, so this is the outline. I want to start. I know that this is a fairly general audience. I think it was widely advertised to lots of folks, both within and outside of science. You have to have a little bit of a science background, or at least a penchant for science, to understand many of the things I'll be talking about. But I want to tell you a little bit about the nanoscale. Talk about why nano. I think that's worth asking. What's the big deal? Why are people so interested in it? In particular, in the context of nanomaterials, and I know there's a very strong effort here in materials, so this is going to be very important for this crowd. Talk about how some of those materials can be used in the area of biology and medicine, and in particular, talk about what nanotechnology can do for medicine in the context of medical diagnostics, in vitro diagnostics, but also new imaging capabilities and ways of tracking what occurs in the body, and this idea of theranostics, the co-delivery of a medicine and a diagnostic in one event. And then I want to finish with kind of a nanotechnologist's approach to learning how to mimic biological systems and to see if we can create a new type of therapy based upon that concept alone. So we're going to talk about a lot in a relatively short period of time, but hopefully we've got a problem with this thing. It's not working.
Is it possible to turn the lights down on the stage to make your lights more visible?
That would be nice. I don't control that. So let's start by talking about how small is a nanometer. So these are often examples of nanostructures, and you can see why nanotechnology might be important in biological systems, because you know, I'm a chemist, and I think in a very chemistry-centric manner, but in reality, this is not the scale of chemistry, it's a scale of biology. But to make an impact, you have to learn how to begin to build materials that interface with biological systems. And that means you have to learn how to do chemical syntheses or to fabricate structures that have length scales on this very tiny length scale, 1 to 100 nanometers. So one of the questions, of course, is how small is this? And I thought one of the best ways to do that would be to show you a clip from a NOVA episode where David Pogue, a New York Times science writer, was interviewing me and trying to tease out some of these issues. And they have, I think, a very nice way of illustrating what scale we're talking about here.
You keep saying you're building things on the nanometer scale. I don't even know what a nanometer is. So this is a meter, this is a centimeter, this is a millimeter. Is this a nanometer? What's a nanometer?
I'm going to try to illustrate higher for you. If we shrink you by a factor of 2, you're about the size of a small child. We continue to shrink you by a factor of 2, 4 more times, you're about the size of a golf ball. We go to 10, you're now the size of an ant. We keep going another 7 times, and now you're about the diameter of a human hair. That's roughly what we can see with the naked eye.
Even after so much shrinking, I'm not even close to the nanoscale. Cut me in half 5 more times, and I'm the size of a red blood cell. 5 more times, I'm a virus. 7 more times, and I'm finally 1 nanometer in size. 1 billionth of a meter. That's less than half the width of DNA.
Okay, so that gives you kind of a feel for scale here. Again, just 2 benchmarks. A red blood cell, which we can see with an optical microscope, is huge on this scale. It's 5,000 nanometers, so 5,000 times a single nanometer. If you think about nanolithography, and we talk about making some of the circuit diagrams that are now made through modern microfabrication and nanofabrication approaches, the line widths, when you see them in magnified form, if you applied the same level of magnification to a line that was drawn with a ballpoint pen, that line would be about a mile wide. So these are really, really tiny structures, and structures that as I said, are small enough to begin to think about interface in biological systems. Now why nano? What's so interesting about this? This is a real materials play. So one of the real exciting things from a scientific perspective is that everything, when miniaturized, is different. And that's why scientists are so interested in this field. So if you take any bulk material and shrink it down to the nanometer length scale, the sub 100 nanometer length scale, it'll have new properties. Take, for example, gold. These are all examples of gold structures here. Tiny particles, we can see them if we do electron microscopy on them. They're not gold in color, at least when they're at this size. You can see they're kind of green in color. You see silver. These are 3 different sizes of silver particles. Again, not silver in color. I can have yellow, kind of a light blue, and a darker blue or gray here. And if I make this go from a spherical particle to a triangular prism, again, all silver, I get a red color. This is looking at the way light scatters from these particles. And so that's kind of interesting, right? So you take one material, you shrink it down, and you get completely new materials depending upon the size and shape. And so what this tells you is that if you really want to exploit this field, you have to really learn how to make these structures on demand, where you can control architecture on this length scale. And you have now fundamentally new properties that you can think about exploiting in the development of new types of technologies. Now, this is not a new concept. Back in the Middle Ages, the ancients recognized this. In fact, if you go look at the cathedrals, Canterbury Cathedral, or go to Florence and look at the beautiful cathedrals there, you'll see these wonderful stained glass windows. Many of the colors from those windows comes from a type of nanotechnology, in the sense that they knew that if they had salts that were exposed to the right types of chemicals, a reducing agent, and the right types of thermal processing, they could create colors that would stand the test of time. And those colors came from tiny particles that were made on the nanoscale to give you the colors that I just showed you. So many of the reds that you see come from gold nanoparticles. And they used materials that had gold salts in them, in the glass precursors, to reduce and stop at that size scale. And the reason they were so attractive was that, unlike the dyes in your clothing, oftentimes organic dyes, they would stand the test of time. They wouldn't photo bleach. right? So, or photooxidize. And so they not only would be brilliant in terms of the color, they would last for centuries. And to this day, people use these types of particles in stained glass windows. And in fact, if you're interested in this, they go through a big, long discussion of this in the Making Things Smaller video in Nova. Okay, so what does this have to do with medicine? I want to tell you a little bit about how you can begin to use these same types of particles that were used for those types of applications now in the development of very powerful new types of diagnostic systems. And not diagnostic systems that are the basis of scientific papers, but diagnostic systems that are now in hospitals and beginning to change the way we perform medicine. And we'll talk about why that is in a little bit. New approaches to using these types of particles to go into animals and ultimately humans so that we can track where different therapeutics go and cause, for example, release events that cause the delivery of a particular therapeutic to treat all sorts of debilitating diseases, including many forms of cancer. And then I'll talk about new drugs that are being developed based upon some of these principles and this ability to manipulate and build structures on this length scale. To do that, though, I have to kind of introduce some important concepts. I think many people know what DNA is, or at least have a feel for it. We know the double helix, right, made up of two strands, two polymeric strands that have bases hanging off them. And that's the code that we always hear about. It's a very simple code, ATGC. And you know that A's match up with T's and G's match up with C. That's another way of saying that these types of bases recognize one another and can hydrogen bond with one another. And you can create then polymers that have a particular code. And if you create another polymer that has a complementary code, they'll recognize one another and form the famous double helix. And so there's a programmability associated with these types of structures. And these types of structures are the basis for life, right? They're the blueprint for life. They encode everything about us. One of the things that's really neat about DNA and RNA, as you'll see in a little bit, is that you can now make these types of strands in the lab. And you can do so over the course of an afternoon using modern gene machine types of techniques. In fact, much of that technology was developed by a colleague of mine named Bob Letzinger, who did all the original organic chemistry to enable the modern day gene machines and allow you to do really much of the chemistry and much of the material science and nanotechnology that I'll be talking about. But one of the ideas we had back in the mid-90s was that we would like to learn how to take particles, the same types of particles I just showed you, for example, a gold particle, and synthesize DNA strands that you could tack onto the surface of the particle. And so we developed strands that had chemistry that would allow us to anchor them onto a particle surface. So, for example, with DNA, we could put a little alkyl thiol end group. It's a sulfur group. Sulfur loves gold, and it could form a bond. And if you added many strands, you could begin to create structures like these. And we like to refer to these as spherical nucleic acids. And this was kind of a new concept because the idea was, and we weren't even thinking about developing medical tools. We said what we'd like to be able to do is create a programmable atom. Kind of look at the nanoparticles in atom and say, could we put different strands of DNA on it and then use base pairing, right, DNA binding schemes to assemble those artificial atoms into complex materials. So you could literally program the formation of any material you'd like from the bottom up, and you could control the placement of any sets of particles you'd like using the DNA base pairing interactions, which any undergraduate and many high school students now know, the ATGC code, to program that. It turns out when you make these types of structures, though, they have some pretty spectacular properties that make them very attractive in the development of powerful new biological and medical tools. And in fact, I like to think of this, when we think about, what we know about DNA types of structures. This is the form that you guys are all familiar with, linear duplex DNA. That's a double helix. It also comes in a circular form. It turns out certain plasmids have circular DNA type architectures. In this type of architecture, we think is kind of a fundamentally new entry in this whole way we view one of the most important molecules ever synthesized by chemists. And you'll see why in a second. So what I'm going to show you is that on a sequence for sequence basis, these types of structures are night and day. They're completely different. And so that's kind of a powerful concept again. You take maybe the most important molecule ever synthesized by chemists, you develop chemistry for modifying nanoparticles, arrange it in the spherical format, and on a sequence for sequence basis, you get something completely different. Okay, what do I mean by that? Well, just look at the ability to bind or recognize a complementary strand. So this is normal DNA. If I add a complementary strand, I can form that double helix. If I look at the same spherical architecture and its ability to bind to that complementary nucleic acid, it binds 100 times more tightly. So it's 100 times better at sponging up the complementary DNA. That turns out to have significant consequences in the area of diagnostics. When it binds, I'm going to show you what are called melting transitions. You can heat DNA, and at a certain temperature, it'll unravel. And that typically occurs for these types of structures over very broad temperature ranges. It turns out with these structures, it occurs over a very narrow temperature range. That turns out to be important in the area of diagnostics because the sharper that transition, the better your ability to differentiate perfectly complementary target strands, for example, from non-complementary strands. And then the other thing that's important is, we call these spherical nucleic acids to focus on the shell because it turns out you can swap out the middle, the core, and make all sorts of different types of spherical nucleic acids with different physical properties. Remember I showed you how the colors changed, how the light scattering properties changed as a function of whether we had gold or silver or different sizes and shapes? All of those can go into these types of architectures. Many of the properties remain, but the physical properties change depending upon what particle core you use. So here's the first result. This goes from, in 1996, I had a really talented grad student, a fearless one, because we'd never worked with DNA before. I told him, you need to go learn how to make DNA, and you need to learn how to tack it on to inorganic nanoparticles. And he was smart enough, he walked up to Bob Letzinger's office, who invented these gene machines, and said, how are we going to do this? And we came up with a scheme for synthesizing oligonucleotides that could be attached to these particles and to create then recognition elements that could add to, for example, a strand like this. So if you look at this, you say, what are we actually doing? We can make two different types of particles that have different DNA sequences on them, and we'll call them A and B. And they're different based upon that code that I was talking about, right? And so think of them as kind of like chemically specific Velcro. I can make them recognize any complementary code that I can also make. So I can synthesize a strand that we'll call A prime, B prime. This portion of it recognizes this. This portion of it recognizes this. And so if this is present, these particles should zip together and form an aggregate type of structure, right? That was that idea of building materials from the bottom up. Well, he did this experiment. His name is Bobby Musick, and he came running down to me. I'll never forget it. He said, it was at 11 o'clock at night. We worked long hours back then because I was just an assistant professor. And he said, you got to see this, Chad. This is amazing. He said, I add the DNA, and I go from this state to this state. Remember I showed you the nano gold has this intense red color? This is really dilute. This is nanomolar, 10 to the minus 9 molar concentration. So it's really, really dilute. You couldn't make a measurement on DNA alone, but you can on the particles here. When you add the DNA strands, you go from this, I think the provost is an Ohio State person, Ohio State red, to Northwestern purple. And what that's doing is amplifying this recognition event. So we're able to watch this formation of duplexes, right? That's imposed by this linker strand being present, and I generate this purple solution. And the reason is when the nanoparticles get close together, they can start interacting with one another. And there's something called a plasmon resonance, and they can feed into one another, which gives rise to a color change in this case. He says, that's kind of neat. He goes, but this is really cool. He takes it and he puts it in a glassware drying oven, just a little oven, heats it, and it goes right back to red. Puts it back on the bench top, and it goes back to blue. And what we were watching with the naked eye was the raveling and unraveling of DNA. Right? So we had a way of amplifying it and presenting it in very simple form. And almost immediately we said, new detection system for DNA. Just kind of kidding but half serious at the same time. And the reason I say that is, when you think about this, you can say, okay, that's nice, I made that strand, but in reality, everything living, whether it's a bug, virus, has a genetic code. And those genetic codes are becoming known through a lot of modern advances in sequencing. And if we know the code for a given type of disease, bacterial or viral, we can begin to create particles that recognize these types of targets and now create very simple detection systems, colorimetric ones, that allow you to probe whether a given target is there. And you can do it, in principle, at very, very high sensitivity to see very low quantities of the target of interest. That actually took us then into this whole area of medical diagnostics. And so again, my roots were in materials. Everybody said, hey, how did you end up here? Again, following your nose, you make a discovery and say, hey, this could be very important in biology and medicine. Let's begin to learn a little bit in those areas and see if we can make a difference there. And so I'm going to show you how these types of particles can be used to enable a new FDA-cleared medical diagnostic system that, as I said, I think will change the way medicine is carried out, because it allows you to do very simple, very sensitive, and very selective detection at the point of care, something that we can't currently do and do very well. I'm also going to tell you about some new systems that allow you to do those types of measurements, but in living cells. So moving beyond in vitro medical diagnostics, where you take a sample of blood, saliva, or urine, but where you can actually go probe what's occurring in a live cell. So, here's the crash course. Every living organism has unique code. That's the good news, right? Different codes can be linked to different genetic, bacterial, or viral-based diseases. So we can say tuberculosis has one type of code, anthrax has a different type of code. I can create then particles that are unique for those particular codes and recognize those codes selectively to give me these types of physical property changes that are the basis then for detection. And with the Human Genome Project, we're identifying all sorts of sequences that are going to be very important for tracking many types of diseases. In fact, all types of diseases that have a genetic basis. And so the question is, how can we rapidly and reliably identify different nucleic acid sequences and diagnose diseases? So I'm going to skip forward a little bit. It turns out that you can use a variant of what I just showed you to do very sensitive and very simple detection using these types of particle probes as high sensitivity indicators. And so what you'd like to be able to do is not just detect one target, one disease target, but detect many different disease targets. And so the way we did that was we created a chip, just a little microscope slide, and we put little spots of DNA on it that could take a sample, blood, saliva, or urine, that's been processed where the DNA targets have been released associated with a given disease target. Let that chip sample the target, find the right target or targets, have it bind to the right quadrant. And so you can see here, in this case, it's binding to this little red quadrant. And then we develop with a nanoparticle probe, and that binds. That gives me a signal. Now, DNA binding is not perfect. It's nice because it's programmable, but if sequences are similar, you still will get particles binding there, and those would give you all false positives. But remember that sharp transition I was telling you about, the melting transition? I can pick a temperature where only the perfectly complementary is on. Well, I can use that to wash away all the mismatched strands. And so only the target that we're after binds, or predominantly binds. And then here's one other trick. It turns out nano gold has a property that bulk gold doesn't have, and that is it will promote the reduction of silver 1 in the presence of hydroquinone. A lot of the non-chemists are going, what the heck does that mean? That's a fancy way of saying it will develop photographic developing solution. So if you take ordinary photographic developing solution and put it over this chip, anywhere where you have the gold particles, you plate out silver. And now if you bring light through the side of the microscope slide, you can measure the color, the scattered light coming off of those silver spots. And that increases your signal by a factor of 100,000 in less than 5 minutes. Okay, so put this all together. Here's a biological weapon simulant type of panel. Hepatitis B, smallpox, Ebola virus, and HIV. All targets that we're trying to detect in a sample. Remember, this is just the DNA, so we don't have to worry about the disease here. We're just looking at the signatures right now. But here you can see how this works. This is a false color scale where we're measuring light scatter from these spots, from the silver spots. And we set this up so that orange or white is large signal, and this blue or black color is low signal. And you can see this is done 6 times over for each target, so every one of these is present. Only a hepatitis B, 1, 1, 1. 2 guys here, 2 here, 2 here, 3, and 3. It's not perfect, right? You see background here, but that's low signal. And you can set a threshold to determine what's a real hit versus a false positive. And you can quantify based upon the presence of these targets. Now, what does that do for you? Well, that becomes the basis for this FDA-cleared diagnostic system I was telling you about. Sold by a company we started about 12 years ago called Nanosphere. And what these guys wanted to do was to take this technology and create some of the first point-of-care medical diagnostic capabilities to go after these types of nucleic acid-based signatures. And to do that, it has to be sensitive, it has to be very selective, you have to be able to detect many things at once, and it has to be very, very simple. And this really had a lot of the hallmark features for doing that. And so you can see these are some of the capabilities. I thought, again, the best way to get an understanding for this guy, David Pogue, he was interviewing me about nanotechnology. He said, I want to go see this company. I'd like to figure out how this technology works. And so he did. And you can, this is truly remarkable. I couldn't have set this up any better. I mean, you see what happens.
Chad Mirkin has developed a technology that harnesses the unique properties of gold and silver nanoparticles to test for genetic variations in patients. Sequencing DNA is expensive and time-consuming, but Chad's revolutionary test takes less than 2 hours. I offered to bleed for science to see how it works.
And your name is?
David,
Okay, so we're doing a low blood draw today. You can relax.
is this the extraction bench?
Yes, this is.
First, a technician extracts pure DNA from my blood sample.
This is my DNA.
This is your DNA.
He then loads it into a small disposable cartridge and inserts it into the machine for testing. This test can actually read the letters of my DNA, and using gold nanoparticles, it flags variations that might make me unusually sensitive to particular drugs, or even mutations that signal heightened risk for disease. Less than 2 hours after drawing my blood, the results are in. It turns out that the test has some interesting news for me about my sensitivity to a blood-thinning drug called warfarin, or Coumadin. It's commonly prescribed to stroke and cardiac patients. It's a potentially life-saving drug, but if the dosage is wrong, it can cause fatal bleeding.
So what's my warfarin dosage?
What they call a double hit, I'm sorry to say.
A double hit?
You're a double hit. So you have two genes that are mutated, and therefore you're very sensitive to warfarin. And your calculated dose is 2.7 milligrams.
See, I knew it. My mother always said it would be like 2.6 or 2.5, but I always said, no, Mom, mine's 2.7.
You should always listen to your mom, David.
The nanosphere test gave me some crucial information.
This is the mutation.
Quickly enough to save my life if I'd been ill. It's a diagnostic tool. The next goal is to fight illness in the body at the same tiny scale.
Okay, so that's pretty amazing if you think about it. So that technology goes from in 2000, it was a science paper, to an FDA-cleared system in 2007. He's back there last year getting tested for these genetic mutations. We didn't know what he was. There was a 1 in 24 chance that he had both those mutations, and he found that out, in real time using that particular assay. And so that's a game changer, because now you can think about not having technology where you have to go to a remote lab. So much of the modern diagnostics industry relies on sending one of your samples of blood to a Quest Diagnostics, for example, off-site. Let them do the test, and a couple days later you get the results. Now you can get the results in less than 2 hours. And so there's one example there. There's a more compelling example, though, that was just recently FDA cleared, and that's in the area of sepsis and bloodstream infection. This is a huge problem, both on the civilian side and the military side of things. So if you get a bloodstream infection, you've got a big problem. There's a decent chance you're going to die from it. They have to be able to diagnose and figure out whether you have that and then put you on the right types of antibiotics as quickly as possible. Right now, what they typically do is if they think you have sepsis, they just load you up with all sorts of antibiotics, including vancomycin, which is the last stage of antibiotics. Big problem, because you don't need that. There's also a big problem in terms of creating resistance, and it's incredibly expensive. And the reason they do that is with the current assays, they have to culture blood up, and it takes 3 days before they can get the results. So now with these types of tests, you not only can figure out whether somebody has sepsis, you can figure out what types of bugs they're infected with and prescribe the right types of antibiotics. And so it's a big game changer in terms of the way we think about doing diagnostics and ultimately treating patients in this regard. And so this is, I think, very exciting in terms of seeing some advances in modern nanotechnology translate then into real medical benefits. But we like to really push the frontiers. And so I'm a big proponent of asking people to look at analytical benchmarks and ask where we can make a difference. And so in the area of DNA detection, there are actually quite a few different types of DNA detection systems out there. I've showed you some of the strengths of this new one. But what you have is extraordinarily high sensitivity, where you can fish out very low concentrations of a given disease marker. Many of the tests that are out there are based upon protein targets. Right? PSA is an example of that. We'll talk about that in a second. And there we don't have the ability to fish them out at very low concentration. And so I challenged our group to come up with nanotechnological methods that would allow us to begin to access this part of the analytical scale with these types of nanostructures, where we can fish out protein markers at really low concentrations and exceed what you can do with conventional ELISA type of technology, which works in this part and up in terms of target concentration. And so why would you want to do this? My view is that if you can detect really minute quantities of a particular protein target associated with a given disease, you could change the way, again, medicine is practiced. We can track disease at earlier states. We can look at recurrence in certain situations. We can look at residual disease. You know, a little bit of cancer is a bad thing. Catching it very early is very important. And this turns out to be very powerful. You can use these types of structures to create very high sensitivity protein tests as well. And I just want to show you one example here that came from a collaboration with one of my students named Shad Faxton, who was also a PK winner. He was with Jeff at meeting the president because he got it through the Air Force. But he was a medical doctor that we brought into the group, and he was a urologist. And we developed these very high sensitivity assays that allow you to track very low concentrations of protein. And so we were looking at where we could really demonstrate the power of these types of technologies. And he said, why don't we develop an ultra-high sensitivity PSA test? And you listen to that and you say, well, for those that are over 40 in the audience, if you're a man, you say, that doesn't make a whole lot of sense because I can go get a PSA test today and the doctor measures my PSA level, and if it rises, they often then say, well, you need to go in and have a biopsy, and if you have a biopsy that's positive, then you have to have it surgically removed or resected. And that's true. But what he thought about was, what would happen if we could increase the sensitivity of this type of test? What new technologies would it open up? And if you think about it, routine screening won't benefit from higher sensitivity because all men have a pretty high level of PSA in them. But we began to ask the question, what happens to a man after his prostate is removed? Many people go through this surgery. What happens to their PSA levels? Can we detect with conventional technologies? And what would happen if we could detect? And it turns out some interesting things are discovered through that process. First of all, it turns out almost everybody post-surgery have 0 levels of PSA in them with the conventional tests. It's not 0, though. It's below what's detectable with the conventional tests. If you use these new types of assays, nanoassays, you can begin to not only track or measure the concentration, but track it over time. And so we did a clinical study with 500 patients these are some representative data sets here where you can see different types of populations. In this case, you can see a person who's at a very low level of PSA. The limit of detection with the conventional technology is called ELISA. It's way up here. Everything down here would be registered as a 0. This person has a low amount of PSA and it stays low for a long period of time. That person has had their prostate removed and they're not going to recur from this disease. Okay? That's good. So you know that person's never going to rise. I don't know who's hitting the switch, but it keeps going up and down. That person is never going to rise to a detectable level with the conventional technology. This person starts out flatlining, and then they start to rise. And eventually, they're going to get detectable with the conventional technology. So you can see the conventional technologies in red here. These are people that started out low, and then they rise. They're recurring. Okay, so roughly 52 to 58% of the people, depending on who you believe, fall in this category. The rest are over here in this category. What do we get from that? Well, you can take half the population, and you can say, take the weight of the world off their shoulders. You can say, you're not going to die from this disease. You are flatlining, and you are effectively cured. These guys aren't so lucky because there is no known therapeutic for them. There are all sorts of experimental therapeutics, but we still have a very important way of helping these people because the first step to a good therapeutic is a good diagnostic. And so the ability to track PSA on a different radar scale here in this case is very important because we can see it earlier and we can watch these rises and we can look at whether they rise or fall, these levels, in response to different types of experimental therapies. And so we think that's going to be the first step towards validating important therapies for these types of folks over here. And so this I think, is going to be a very important set of tools that go in tandem with efforts to develop new types of therapeutic capabilities for these and related diseases. But it shows you how being able to track residual disease is quite important here. And in fact, some of you might be saying, well, people argue about whether PSA is a good marker for prostate cancer. That's true. There might be some people in the medical community for routine screening. But if you have a prostatectomy and your PSA levels drop to below detectable, if they rise again, it's unambiguous that you're recurring. Okay, so it actually turns out to take a marker that's not very good for routine screening and translates it into, or arguably not very good for routine screening, translates it into a great marker for looking at recurrence. This type of story, though, I think is going to go recur and recur over and over and over again. Here's an example. With Alzheimer's disease, we had people, after they saw us publish on the high sensitivity of these types of assays, they said, you know, why can't we create a molecular diagnostic for Alzheimer's disease? Why can't we track something in the blood that's associated with the disease? And here's the problem. There are lots of known markers for Alzheimer's disease at relatively high concentrations in the brain. But when you move away from the brain to cerebral spinal fluid and then to blood, the concentration of those markers drop off precipitously. And so we're stuck right now in this area for, first of all, not having good ways of diagnosing the disease, period, and certainly not very good ways of treating it. Lots of money is spent on this every year. The only unambiguous way of diagnosing someone with the disease is to do a postmortem analysis of the brain. That is a low bar in terms of developing a diagnostic, okay? I had a colleague, Bill Klein, who came in and he said, look, we have some markers, new markers. They're called ADLs, and they're little protein bundles. They're actually nanostructures themselves. And he believes that these markers are at relatively high, he knows they're at relatively high concentration in the brain. He can image them. He can also create tests for them based upon samples from brains where he can detect them. But if he moved to cerebral spinal fluid or blood, they register effectively as zeros with the conventional technologies. And so the question was, could you use these high sensitivity assays to begin to track this disease in new ways? And so again, I'll just show you some sample data here. These are examples of patients that we've looked at. Samples of cerebral spinal fluid, and we're measuring ADL concentration. And the concentration is femtomolar for the aficionados, very, very low here. You can see these people who have the disease are scattered all over the map. Every one but two are statistically elevated. They have high levels of ADL in them, and the amount they have depends upon the progression of the disease. The control population, an aged population that does not have the disease, they're all clustered around this very low concentration, which you could not pick up with a conventional diagnostic tool. And so again, what this points to is a way of using, again, markers, in this case studies of histological samples of the brain, to begin to create new types of diagnostics that are relevant, moving away from the brain, in this case to samples of cerebral spinal fluid, and hopefully ultimately to blood. Okay, so those are examples of you know, how you begin to think about changing medical diagnostics. What about other areas? Here are some other properties of these spherical structures that I want to point to. Again, linear DNA and RNA will not enter cells very easily. And there's a whole field that focuses on trying to move these types of structures into cells, because if you can move them into cells, you can begin to think about doing some interesting things with them, both in the context of detection and the development of therapeutics. These types of structures, it turns out, naturally enter cells. You can feed them to cells and they pick them up quite readily. And I'll just show you that right here. This is really remarkable. You look at cells, these are mouse blood vessel cells, treated with these types of SNA gold nanoparticle structures. You can see the cells are turning almost black in color because they're picking up millions of these particles. This is not just a nano effect. It's not just because they're small. You look at the same cells over here treated with particles that have a protein coat instead of this DNA coat, and they just stare at them. They stay white in color. Okay? This maybe illustrates it even better. Maybe if you turn the lights down a little bit, it'd help. This is what I was showing you. If I take a DNA and I put a fluorophore on it, something that'll light up when it goes inside the cell, you can see here there is no evidence of that fluorophore being present. These are the same DNA strands arranged on the surface of the particle. 1/100th the concentration, so 100 times more dilute. I feed them to the same cells, and the cells suck them up very rapidly. So now what you have is the ability to, again, you take this very simple molecule, it shows you the importance of structure, you arrange it in the spherical format, and you get a structure that naturally enters cells. This turns out to be quite general. It goes into almost all types of cells that we've studied. Nerve cells, you can see skin cells, things called primary cells. Tissue samples will pick them up. I'm going to show you organs that will pick them up quite readily. And so what that means is you can think about doing all sorts of interesting things now, not outside the body, but inside cells and inside the body. Okay? So one of the things that we've developed is a structure that we call a nanoflare. And what we wanted to ask was, if we could get particles inside cells, could we begin to measure what's inside them? Could we begin to get signals that would correlate with the concentration of different types of targets inside cells? And we'll talk about why that's important in a second. And so what we created was a structure like this. It has that spherical arrangement of DNA, and then we have a short strand of DNA with a fluorophore, that thing that lights up, next to the gold particle. And the reason is that gold acts as a switch, and it turns off the fluorescence, the color of that fluorophore. When this goes into a cell, the idea is we can design it so the end of the strand can recognize a target mRNA sequence, and we'll talk about why that's important in a second. When it recognizes it, it can bind to the entire sequence here and peel off these short oligonucleotides, these short strands of DNA with fluorophore. And when that moves away from the gold particle, it's going to turn on fluorescence. And so if you think about this, I can create particles that selectively recognize these types of targets. Okay? So here's the idea. Cells are different based upon these different genetic signatures. And so maybe what I can do is begin to create nanoflare architectures that can go in and measure and differentiate, measure the concentrations of these mRNA targets and differentiate, for example, cancer cells from healthy cells. And so the idea is you could create flares that go in and recognize mRNA sequences in cancer cells that are elevated compared to their normal cell counterparts. And so in this case, I would get a cell that turns bright red like I showed you, and in this case, I would get one that does not. Well, it actually works beautifully. So here you can see examples now. These are cancer cells, SKBR3 cells. You can see them lighting up. They're breast cancer cells. Red fluorescence we're going after a target for the aficionados called survivin. It's unique to these types of cells, or elevated in these types of cells. And then these are normal cells where you see barely any fluorescence. Now, what does that do for you? Well, here's what it opens up. This is the only way, there is no other way known, to go in and measure genetic content in live cells. So it changes what you think you can do about, or what you can do with these types of technologies and how, and what you can develop from them. So what we're doing now is beginning to create suspension assays where we can take a sample of cells where we might suspect, for example, in the case of breast cancer, you have circulating tumor cells. If there's some cells that are circulating in the bloodstream, a small population of them, I can feed all the cells these types of nanoflares, and then I can fire them through something called a flow cytometer. And I can tell that instrument to differentiate these based upon fluorescence. So I can literally count the number of cancer cells that are circulating. And that could create the new ways of diagnosing important diseases and debilitating diseases like breast cancer. And so we've just begun to do that, where we're now taking, this is volunteer blood sample, that's translation Chad Thaxton's blood. He's the assistant professor that works with us. He bleeds for society regularly. We spike these with breast cancer cells, and then we use the nanoflare technology to again, feed to the entire population. And these are the types of plots that you get. And using this technology now, we can literally pick out as few as 10 cancer cells in an entire sample of blood. And I think this is going to be used heavily to develop all sorts of new diagnostic screening tools, and again, translate this capability of cellular entry into powerful new medical diagnostic tools. Okay, what about therapeutics? Is there a way of doing something with these types of structures that will change the way we develop new therapeutics? Well, this is important, this idea of being able to get particles into cells and to latch on to genetic material, because you can think about creating what's called gene regulation therapies, ways of getting particles into cells and then flipping genetic switches associated with a given disease so that you can turn them up, turn them down, or turn them off. And for example, in the case of cancer cells, you can go after genes that cause them to selectively die in the presence of healthy cells. And here's an example of where nanotech plays a huge role here. So, I've introduced the spherical nucleic acid architecture. This changes the whole way people think about developing therapeutics in this field. I think you guys had Bob Langer here as a Ford lecturer about 5 years ago. He goes down this particular path here. Everybody in the field does. The lore in the field is that you must use positively charged polymeric carriers or viruses to trick cells into picking up nucleic acids. And what I just showed you is that if you arrange these in spherical format, they naturally go into cells and you don't need a co-carrier. Well, those polymeric carriers that they use are great because they deliver, but they also cause all sorts of toxic side effects and problems. And so the ability to deliver these directly is potentially a very large advantage in the development of new types of gene regulation therapeutics. And so we've begun to explore this. And so I'll just give you a feel for how this works. So this is a crash course in regulating things using a gene regulation approach. One way to do this is via what's called antisense pathways. And the idea is, so remember I said DNA is a blueprint for a cell. It creates a message called messenger RNA. That then goes to something called the ribosome, which is a factory that reads the message and translates it and turns it into a protein. And then these proteins are used as the basic building blocks for almost everything, all tissues and what makes us us and what makes cancer cancer. So the idea here is that if I could somehow go in and selectively knock out the messages associated with a particular type of cancer or disease, I could stop the production of the disease-causing proteins. And so here's how we do it. The idea is if you can get particles to go in, you can design them, and I've already shown you they will go in, to target a particular mRNA sequence. That'll bind and block the ability for this to get to the ribosome, cause it to be chewed up, it turns out, and that effectively stops in the production selectively of certain types of proteins. And so one thing that you can target in the case of cancer, what differentiates many cancer cells from normal cells is that they are immortal. They live forever. And they produce proteins that stop what's called apoptosis. So the idea is if you can target those types of proteins and stop them from producing those, they will die like normal cells. Here's the kind of neat both plus and minus of what I've described so far. Turns out these particles, I showed you, they go into almost all cells, all but mature red blood cells. They go into everything. So if we pump them into animals and look at the distribution of the particles, they go into all of these different organs. And that's scary in one respect, but exciting in another. Because it turns out it's hard to get therapeutics to certain locations, especially, for example, the brain. You have to be able to cross what's called the blood-brain barrier, and that's not a trivial thing. About 1% of what we give to an animal ends up in the brain. That turns out to be a reasonable therapeutic dose. The other thing that's interesting is they'll go into not only these types of organs, they'll go into the largest organ in the body, which is the skin. So here, if you look at this, this is a perfect application for something that doesn't have selectivity with respect to organ penetration. I can apply it to skin locally, and the skin's very difficult to get things across because it has all sorts of barriers, including something called the stratum corneum that blocks the transport of materials, which is good, right? That keeps a lot of foreign elements out. But these types of structures, they're labeled here in red, go through the skin quite readily. And that opens up now the ability to begin to target genes in the skin. And here you can see we're targeting something called EGFR, which results in a thinning of the skin. And you can, in this case, these are mouse skin samples. You can see skin that's been treated with particles that have the wrong types of genetic constructs show no effect, whereas you can see a significant thinning effect over here. Why is that important? Well, now that opens up the ability to create all sorts of new therapies for diseases like melanoma and other skin cancers, psoriasis, keratosis, and you can even think about cosmetic applications where you can up and down regulate genes that change anything from pigmentation to elasticity of skin. And so this is going to open up a whole new area and take nanotechnology down a completely new path because of this unique ability to get these types of structures into skin. And so here you can see conceptually what we're talking about. You can put these in creams, ordinary things like Aquaphor, this is my daughter's hand, and apply them to localized areas of the skin and move these types of constructs in and flip these genetic switches associated with a given disease. What about more aggressive diseases? This is where things are really exciting. It turns out that they'll move through all sorts of tissues, including the brain. And so one of the experts at Northwestern, the medical school, a guy named Dr. Alex Steg, came to us and said, if these things really move through tissues like you say they do, they'd be fantastic to begin to create new types of therapeutic approaches for brain cancer, glioblastoma, multiforme. one of the most aggressive forms of cancer out there. If somebody gets diagnosed with this disease, typically, even after surgery, after they remove the tumor, you have 12 to 15 months to live. And so the hope was that we could learn how to give these types of structures, either locally or inject them into the bloodstream, to create new types of therapies for this disease. And the idea is that if you could get them to the brain, you could begin to flip genetic switches associated with those cancer cells and get the few cells that are left behind when a surgeon removes a tumor, and hopefully stop the disease dead in its tracks. Well, this shows you, again, the power of these types of technologies. You can see here, we have great imaging facilities at Northwestern as well. Tom Mead always likes me to show this because it's from his facility. I know some of you guys know Tom. This is a whole mouse brain that we treat with these types of constructs, and we put little labeling agents on them, gadolinium groups, that allow us to track the movement of the particles inside the brain. And what we're looking at is a point injection of the particles. And normally, what Alex would tell you is that these things stay very localized. But here you're going to see, after 24 hours, how much of the brain it accesses. And here you can see roughly, this is the path taken by the particles, 22% of the brain. And so what that told us was that if we could get these types of particles to the brain, we could begin to think about treating this very aggressive form of cancer. And so very recently, in fact, I was just on a phone call with the FDA today to talk about filing a new drug application. We've discovered that, originally we were thinking what we're going to have to do is locally deliver these. After a surgeon removes a tumor, we're going to flood the area with these types of particles and try to get the last few cells that are left behind. But we did one more experiment. We said, let's take mice and do a tail vein injection and ask, do these get to the brain? And the answer, surprisingly, was yes. And about 1% of the drug that you deliver gets to the brain. Then we targeted genetic markers associated with the disease that are supposed to do two things. One, if you knock them down the way I described, you can facilitate apoptosis or cell death, and you can also sensitize the animal to treatment with chemotherapeutics, something called TMZ. And here you can see what are called survival curves. These are animals that have tumors in them, surgically implanted, and we've treated, in this case, with active drug molecule. And in these cases, we have no treatment or the wrong types of constructs. And the separation here is what's important. What you can see is that the animals, this is time, the animals live longer if they've been treated with these types of constructs. And so that's extremely exciting for a couple of reasons. First of all, you have a systemic way of treating a very, very bad disease in this case. Second, we had no idea what the dose should be here. This has actually been reproduced now 3 or 4 times. And we always get this consistent response here. And we've been able to actually increase the distance by optimizing the dose. And we're now coupling with chemotherapeutics to see if we can get even further distances between these. But the exciting thing here is we now have the basis for a new type of therapeutic for one of the worst diseases out there. And we're headed towards human trials with this early next year. And we're hopeful that we're going to be able to show that this result of being able to systemically inject these into mice and the ability to cross the blood-brain barrier and localize these types of SNA constructs in the brain translates to humans. Because in all cases, those that have the disease have to have their tumors surgically removed, and we'll be able to measure the gold content inside the tumor to figure out how much drug actually got there. And so what that gives you then is a new way of detecting disease, detecting metastatic cancer cells, and new types of therapeutic leads based upon gene regulation. The last thing I want to show you, though, is a kind of a different approach to developing drugs. So there are 2 ways you can think about creating drugs. One is to create a molecular drug that alters a biochemical pathway, or you can create something that's kind of like a nutrient, a synthetic equivalent. And so I mentioned this guy, Shad Thaxton, the medical doctor that joined, actually the first medical doctor to join my group. He came in and began to look at some of the things that we were doing, and we started talking about other types of drugs that had been developed. And one that's really quite important that many of you are familiar with is Lipitor. It's the biggest drug ever invented. It's a statin. It allows you to lower what's called the bad cholesterol, LDL. But if you have cholesterol problems, like I do, my doctor tells me, he said, you not only have to have a low LDL level, so you need to take a statin, which I do, you need to raise your HDL. And the only way to raise your HDL is to exercise a lot, which I try to do, and to take something like niacin, which will moderately raise it. Much of it's genetic. Chad came along and he said, you know, what's interesting about HDL is that it's a nanostructure itself. And in fact, we can think about reconstituting it. So let me just get to. It is a structure that is made up of lipids and a protein called APO1A. And it typically is right here on this length scale. You can see between 5 and 10 or so, or 12 nanometers. And it's a structure that's pretty simple, a spherical structure. It's got lipids, a hollow core that it can plug cholesterol into, and a protein called apolipoprotein A1. And what we thought about was we said, hey, we could take nanostructures, the same gold particles that I showed you, and we could use those as templates to reassemble HDL on the surface of the particle. And we could begin to create structures that begin to mimic the properties of natural HDL. So instead of creating a drug that would cause the body to produce more HDL, we could create a nanotechnological synthetic equivalent, and you could add then whatever you want, either in pill form or systemically inject it. And it turns out this actually works. You can begin to make structures that when you put them in, first of all, in a test tube, they'll bind cholesterol. If you pump them into animals, they'll bind cholesterol and transport them out. You can see very large concentrations in the adrenal glands, and eventually you can see that the animals excrete these types of entities. And this then becomes the basis for an entirely new way of thinking about drug development. And so we're now beginning to build all sorts of different constructs, looking at how the size and the shape and the composition of these structures changes their activity in vivo. And we're hopeful that this is going to translate then into new types of therapeutic tools. If you look at other types of structures that can be made, Sam Stoop at our place, he's a big fan of making these things called peptide nanotubes. He takes the constituents of proteins and he's learned how to get them to self-assemble into fibrous structures like the one shown here. These turn out to be fantastic nanostructures that can begin to facilitate all sorts of things, including neuronal growth stimulation. And he's been able to take, and I'm just going to show you this, mice that have spinal cord damage and inject these fibers into them and begin to facilitate spinal cord repair. And you can see here 2 videos. This is a mouse. It's not been treated with these types of peptide nanotubes. This guy has. And what these structures do is they create a scaffold that's injectable that allows neurons to grow and repair spinal cord damage. And so there are lots of examples of where you can begin to create these synthetic equivalents using nanotechnology that have, you know, the potential to make a very large impact in a lot of different disease areas. And then I wanted to show you one last example because I think it really shows how far this can go. Jody Simone, who's just a spectacular chemist and engineer at North Carolina and NC State, has come up with an interesting technology he calls PRINT that basically takes tools from the semiconductor industry, the tools that are used to make your computer chips and all of the microelectronic devices, and where he can create then patterns that act as molds to build all sorts of structures out of biologically active material of any shape and size that you'd like. He can effectively take a flu virus, for example, and create a mold of it, and then duplicate it in the form of a certain type of polymer that fills the mold. And you say, you know, what does that do for you? Well, first of all, they've come up with ways of not only doing this in an experimental scale, they can do this in a roll-to-roll process where they can make very, very large quantities of these. And what that means is you can take all sorts of polymeric materials and biologically active materials and adjust a lot of the important properties, the size, the modulus, the shape, the composition, and even the surface functionality. And you can see, you know, look at these. There are all sorts of interesting things from cubes to wing nuts. I'm going to show you some interesting examples here with boomerangs. I think. Okay. So, you know, what he's shown is you can begin to make little nanoscale and microscale particles of any shape you'd like. And if you look at how these types of particles move in different environments, shape and size matters. And so here you can see he's designing structures that kind of mimic pollen particles. And it controls how they rotate in a given plane. So I hope I can get this one here. So you can see those types of structures moving, right, as a function of shape. Here are structures that are called V boomerangs, and you can see they move very differently. Right, they're kind of like the structures that fall off trees, right? Little propellers. Right, so by controlling then not only the composition, but the size and the shape, you can control how these things move in different types of environments. You say, what is that going to do? Well, look at aerosols and how different drugs are delivered. This is an example of a typical type of commercial aerosol where you might be delivering a drug to the lungs. Not much shape control there, right? They now can begin to adjust and make these same materials in any sort of architecture that they'd like. And then they can begin to look at how these types of particles penetrate the lungs and how different types of drugs are delivered. So instead of taking what nature gives you, you can design from the bottom up any sort of architecture you'd like that maximizes the localization of a given therapeutic in this particular application. So I think it's just a nice example of how this type of architectural control can lead to new capabilities. And again, something you wouldn't even think about unless you had this ability to fabricate in mass on the nanoscale. Okay, so that kind of gives you a feel for some of the things that I think are important in coming out of these areas, from diagnostics to imaging to therapeutics. I mean, the message is pretty simple. The challenges here are undoubtedly large. These are really big problems that we're trying to address. I do believe the route is through small technology, this idea of taking structures and miniaturizing them and getting structures with new properties and new capabilities is quite important. And, when this all started, a lot of people said, when are we going to start seeing some of the benefits of nanotechnology? I think people have to start opening their eyes. There are examples. You don't have to say it's all coming. There are many examples of diagnostic systems now that are in hospitals all over the world. Soon will be in community hospitals all over towns that you guys all live in. And I think we're just going to begin to start to see the development of new types of therapeutic capabilities based upon some of these advances. But the timeline there is much slower than on the diagnostic front, so that will always lag the development of diagnostic tools. I want to finish by acknowledging my collaborators. I've mentioned Alex Stegg. Amy Paller is actually a dermatologist. She was very important in the skin work. Shad Thaxton was a student, got his PhD with me. I actually was training to be an MD. He liked the research so much, he bagged the MD, at least the clinical part of it, and took a professorship at Northwestern in research and has been just doing great things. He was the PCASE award winner that I was talking about. And then colleagues that provided examples that I showed, Sam Stoop, Vinayak Dravid, Tom Mead, and of course Jody Simone. This is our institute headquarters at Northwestern, and this is the research group, my research group that did much of the work that I've been talking about. I'll stop there, and I'd be happy to answer any questions if there are any.
So please join us outside. We're going to have a reception. If you have questions for Dr. Mirkin, you can ask him then. Before you go, though, I'd like to present you with a memento of your day here at Case. One of our traditions in the Ford Lecture is to give our lecturers an original piece of art, and this is from Erica Weiss from the Juma Gallery in Little Italy, just down the street.
Fantastic.
And keep us in
it.
your memories. So
I will.
Thank you all, and join us outside. We'll have this